get started, it is my great pleasure to be hosting John Mckay. It was hilarious because, John is one of my oldest friends in the business, colleagues in the business. We met when we were graduate students at UC Davis. He was not a graduate student at UC Davis. He was squatting with the rest of us graduate students at UC Davis. So I've known John for 25 years, something like that. But I didn't even put his name in the hat to come. Instead, Jordan surprised me one day and said, "I'd like to invite John Mckay to be a speaker." And I said, "That's a great idea." So John has worked at the intersection of agriculture, evolutionary genetics, statistical genetics, and conservation. Can I add that to the list? For a really long time. And his career and career are quite broad. They encompass things such as statistical genetics in rice, in Arabidopsis, as well as things like entrepreneurship and starting a breeding company. So I'm really looking forward to John's talk. I hope you all have enjoyed talking with him. He's a really approachable character. If you haven't signed up to meet with him, you want to talk with him, come to tea. We're going to do a tea time. I think it's practical.
All right. Thank you very much. Thanks for having me here. Yeah, I'll tell you what I mean when I say pan-genome, and hopefully we can all agree on what the ideas and theory behind the genetics of adaptation are. So, yeah, so we've been working a lot in crop systems. Some of the advantages of those is there are machines built that can put thousands of plots out for you, and phenotyping is relatively easy compared to even Arabidopsis. So this is my lab group from the last picture from the fall. Okay, so basically the conceptual unification behind my work is this idea of G by E, or this phenomenon, and trying to understand it. So we'll start with this simple cartoon of a reaction norm, where we have two genotypes, a solid line genotype and a dashed line genotype. And the solid line genotype has the highest fitness potential, but probably also the highest resource use. And it's very sensitive to reduction in resources, whereas the gray dashed line genotype is less sensitive to the environment. And so at the trait level, we want to know what are the traits underlying this sensing and responding to the environment, to adjust and buffer the effects of the environment on fitness. And then at the gene level, the same question: where in the genome, what gene models would be polymorphic and underlying this variation in sensing and responding to the environment? So I took the liberty of making up a story in this cartoon that is the amount of roots in this gray genotype that is sensing and responding this limited resource, and basically able to maintain a higher yield or fitness under this low resource environment by readjusting its body plan and in an adaptive plastic manner. So this phenomenon where genotypes express different phenotypes, including fitness, is very well studied in quantitative genetics, and much less considered in population genetics. And so if we think about this this conditional nature of the effect of allelic variation, then you're basically going to have parts of the species range where the selection coefficient on mutations differ, right? So in population genetics, one way to think about things is the amount of time things take to happen, alleles take to go to fixation, and under the sort of global fitness model where mutations have a single phenotype assigned to them regardless of the environment, it's pretty straightforward to distinguish between a selective process and a neutral process. But if sometimes, if in some parts of the species range or some seasons in the across generations, there's positive selection and other times it's neutral, you're going to get a much more muddy picture in between these two, and it's much more difficult to then use things like genome scans to actually find loci that underlie local adaptation as opposed to global adaptation. So this hasn't been completely ignored in population genetics. So sort of the inverse of what I was just saying was modeled by Ota. And the idea here is that if the environment is uniform and selection coefficients are uniform, then the likelihood of fixation and the time to fixation decreases, the likelihood goes up and the time decreases, right? But the opposite of that, if you have heterogeneous environments, with different adaptive peaks and conditional effects of alleles, then the likelihood that any mutation is actually going to go to fixation across the species range is very low. So tying this together then, what I'll show in some examples here in a couple of different species is this QTL by environment interaction, so individual mutations whose phenotypic effect on fitness and other traits is conditional on the environment. We see either trade-offs or conditional neutrality where the allele isn't advantageous to the species as a whole. And in this figure here is an example of conditional neutrality where in one part of the species range, the two alleles have a very large difference in fitness and no difference in other parts of the species range. So obviously, the selection and the patterns that would emerge in allele frequencies across the genome are going to vary drastically between these two scenarios. And what we're typically getting in any species, basically besides humans, where we don't have a whole field of people studying the history of the populations, is that we can't draw these lines of where these clients are, right? So we get this mixed pattern that we try to interpret as soft sweeps or other things. Okay, so I'm going to give some examples in that framework, talking about some empirical data in Arabidopsis thaliana, and then move into thinking beyond allelic variation to copy number variation across genomes and how they might contribute to these quantitative traits that are sensitive to the environment, and then an attempt to try to actually exploit this or make use of it in a crop. So I'll start with the Arabidopsis stuff. This is a project that took place over about a 10-year period. And the idea was basically very classical questions in the genetics of adaptation and to some degree in population genetics. So how many loci can we map that underlie fitness and survival and other traits? And then in this model system, what actually are those genes? What do they do? And then the question I kind of highlighted before is when you have this conditional dependence, then how can you actually find the places? What methods actually capture places in the genome that did go increase in frequency or change in frequency due to this adaptation and cause local adaptation? And then are these, at the allele level, are these new mutations? Is this a mutation-limited process, or are we using standing variation like has been demonstrated in sticklebacks and many other things? So just to cut to the system, this is a system I inherited from my colleagues Doug Shemsky and Yon Ogren. And basically, this is classic reciprocal transplant approaches that are done in plants. So there's populations of Arabidopsis in Italy and Sweden. In this case, these are just the intact local genomes in black and white here. And what you can see at the intact genome level is in most years, there's evidence of local adaptation. So the Italian genotype has higher fitness, and those lines cross, and the Swedish genotype has higher fitness in its local location in Sweden. But there are years, these are the same genotypes and the same physical locations on Earth, but the environment, the climate differs from year to year. And certain years, there's no local adaptation. So it doesn't really matter whether your DNA is the Swedish genotype or the Italian genotype. So this is already an example that with this conditional effect of the environment, selection is much weaker than it would be if there was a consistent crossing over of these reaction norms. So we then, the goal was to then dissect that trade-off at the whole genome level into the components underlying it at the genetic level. Back to the old-fashioned method of making crosses and inducing recombination. So we made recombinant inbred lines of these locally adapted populations, and then planted those same genotypes out in the field over multiple years. So we're putting the same exact genotypes out every year and then measuring survival and fecundity. And between all the arguments, the sample size just kept getting bigger. A nice data set to understand the role of variation in the environment in local adaptation. So if you don't like staring at genome scan continuous data, don't look on the top left. But basically we have this, in this case, survival times fitness for a few hundred genotypes, and then we're doing a QTL approach to understand where the variance in that trait is mapping to. This table on the bottom left is basically testing for QTL by environment, so QTL by site. And we see at almost all of the QTL that there is a significant effect of this interaction. So again, that means the phenotypic effect, and since our phenotype is fitness, the selection acting at these places in the genome is very different across geographies. And since we repeated this over time, and the logic of repeating it was how, if selection and therefore the genetic basis of variation in fitness varies from year to year, how many years do you have to do this in order to actually capture the distribution of QTL underlying it? In this case, the exact answer is 5 years, but I don't know if that's generally applicable. But you can see in the genetic basis of adaptation in the top figure is basically differs depending on the environment, right? So at the QTL level, with the power we had in this experiment, we were able to map 11 QTL that were affecting fitness across these years. And then in this figure, we're dividing them into the two G by E scenarios. The top 4 panels show the trade-off resolving to the level of individual QTL where you have crossover and the local allele has higher fitness in each case. But most of the QTL we found show this pattern of conditional neutrality over the data set we collected, where there's a strong significant effect of allelic variation in one location, and it doesn't matter in another location. So again, if you think about how selection is acting across the species range and across the genome, this is going to give you a much more neutral pattern than the global adaptation scenario that is most commonly modeled in population genetics. So that's basically the take home from the mapping fitness on this, is that we mostly find this conditionally neutral pattern, which is going to give us a more weak population genetic signal than even the balancing selection scenario there, where you have opposite but consistent effects of selection in each location. So one analysis we did to think about that is basically do these genome-wide scans in the context of already knowing the genetic basis of adaptation here. So, we looked at LD, we looked at selective sweeps, and then looked at a couple of associations between allele frequencies and climate. And finally, the workhorse of FST at the top here. And the FST was predictive in a couple of cases, but really not much else was. The sweeps, for the most part, we think are much older than any of the phenotypic and fitness adaptation we're seeing. We have no idea what the phenotypic consequences of these sweeps were. And no one else in the Arabidopsis community has figured out sort of what these. They're pretty repeatable, actually, across different parts of the species range, but we don't know. They're not associated with any phenotypic variation that anyone has shown. We do have, we have some of these major trade-off QTL, where you have alleles that increase fitness at the home location and decrease it away. So since this is a model system, we were able to resolve that to the gene model level by basically editing alleles in each of the backgrounds, and showed that it's the single gene, this transcription factor called CBF2, underlies some portion of the variation in fitness, and particularly for this trade-off QTL, where there's a cost of being cold tolerant in warm places. So this all is, to me, started piquing my interest in this idea of gene duplication and redundancy, because it turns out that CBF2 is in a cluster of a repeat of 3 that happened either right at the Thaliana speciation event or close to it. There's multiple copies in many species, but this array of 3 is unique here. And what we saw in this Italian population is an adaptive loss of function mutation. So all 3 of these genes at the functional level are very similar. They have a binding domain and an activation domain, like many transcription factors. And we see that we're getting this loss of function of activation, but still binding in these alleles. The one gene model is highly conserved in the species, and this CBF of these 3 is the most polymorphic, and most of the mutations end up causing a loss of function of activation domain. So we'll now continue on this theme of thinking about how special this case of gene duplication followed by loss of function might be for adaptation. Pan-Genomes, as in the title, the idea is to some degree or another, we now have these de novo assemblies and annotations of many individuals within a species. And it turns out they have a lot of different stuff in there, not just different alleles, but they have the presence and absence of different genes and different numbers of those genes. And then, looking into this idea of gene duplication, again, in population genetics, the amount of gene duplication and redundancy that we see empirically now was not predicted in the birth of theoretical population genetics. The idea was that if you had a second copy, it was going to be extremely deleterious and go away, right? And some of that work was picked up by Lynch when he was working here, and came up with this idea of subfunctionalization as a way mechanism to maintain some of these duplications and have them last longer. But the idea that they actually could be good, I'd say, has not been considered much theoretically. And you all work on different systems here, but this whole genome duplication followed by loss thing is, it has occurred in all lineages, but it's kind of insanely abundant in plants, right? So here we're comparing mammals to a few different lineages of plants, and the idea is that the top figure is less complicated than the bottom figure, right? So we can actually figure out synteny and one-to-one orthology in these well-funded, these HHMI, NIH well-funded systems, where there certainly is gene duplication and interesting phenotypes associated with it, but it's kind of just an add-on case study thing. Nobody cares about it at the whole genome level. So that's my mission, is to get people like that to care about this. So here is, so you can see that you could guess that the sort of one-to-one orthology is much greater in the mammal system, but here's a table showing that. So, so here are our well-funded systems on the top, and then right now we're doing a lot of work in maize. And so the difference between these two maize genotypes is the same as human and chicken, right? And they diverged less than 1 million years ago, probably less than 8,000 years ago, probably. And because there's whole genome duplications followed by loss, that just scrambles everything up. So here's a picture of that. This is representing one-to-one orthology, including synteny, and starting with aligning everything against the rice genomes. It's not, you can see where the stars are on this phylogeny. Those are recent whole genome duplication events. And here in the maize system, it's pretty crazy, right? These genomes between two individuals are, again, as different as a human and a chicken, but you can cross them. So that's what we're doing, is we're crossing these and trying to figure out what all this stuff does in terms of the phenotype, because so far, this information is new, right? So people have been crossing maize and thinking it's allelic variation, but and there's some of that for sure. We're trying to figure out how much phenotypic variation is due to copy number presence absence, and how much is due to old-fashioned allelic variation. So one way we can do that is actually using all the existing data that's out there in terms of genotype to phenotype. And then, this is one example of many groups that have been making these resources of newly assembled and annotated genomes. Of course, there'll be one that comes out that's better, and someday we'll have the final answer, but we can use these as they are. So there's lots of QTL studies in maize. This is one of them using this extremely large population with 5,000 offspring, and they mapped a QTL for a drought tolerance trait here. And the region of the genome is shown in this riparian plot on the bottom left here. There's a pretty large, what looks like an inversion, maybe this wrinkle. That's a presence absence difference of dozens of genes. And when we zoom in then on, in this case, it's a multi-parent mapping population, but like it's always the case, there's only one parent that actually causes the phenotype at the QTL. And this are basically haplotypes, and you can see that this genotype, this inbred line that confers this adaptive trait, has a huge piece of DNA that has many, many genes in it that no other assembled annotated maize line has. And it's missing a nearby non-orthologous region that everyone else has, right? So there's a big difference in what is in there. In this case, there's no recombination to dissect this further, but the original publication basically said they went to the one reference genome and said, we found the gene. That gene doesn't even exist in the parent that causes it, right? That paralog of that gene model. So we started doing this with our own data as well. I'll show you some of our obsession with root phenotyping in a minute or two, but we started doing all this root phenotyping as in maize and mapping variation in the genome. We did what everyone else was doing, which is look in the reference and find, look at the GFF file and figure out what's there. And there's pretty good resolution in this approach in maize. So we said, oh, there's amino acid polymorphism segregating at this lipase allele. Luckily, we didn't write that many sentences about why it was so obvious that lipase would cause the phenotype. And then we started looking at these new assemblies and annotations and saw that some genotypes have a small bit. This is possibly the smallest gene in the maize genome. So it's a small bit of a transcription factor that is present in some lineages and absent in others, and was not annotated in the reference genomes previously. So we had this. There's no other genes that were annotated. So we got GWAS2. We found the nearest gene and talked about that. But then when we discovered this, then what we're doing basically is manually adding markers around these new things and using genetic mapping, because we need to know, are they really there? So we can use linkage to figure that out, and then basically ask whether these new, whether these insertions, and whether it's an insertion or deletion, I don't actually know, but this copy number that didn't exist in the reference is actually more associated with the phenotype than what we thought was there previously. And so we started doing this basically one QTL at a time, and then thought, maybe we should do this in a more genome-wide way. So, but just to go back to the thinking about the importance of presence and absence variation in evolution and population genetics, it wasn't completely ignored. It was just ignored by the sort of mainstream. So early on, even before there was really a lot of data on the abundance of gene duplications, one of the researchers, so Ono was one of the researchers that came up with a number of explanations of why this actually could be underlying adaptation. And in this case, it's actually more of a dominance situation. So the idea is if having if the genetic basis of heterosis is just having two different versions of an allele, then you could also achieve that just by having paralogs that have different alleles, right? And there's some now, case studies supporting this idea. but yeah, not a lot of because these are not allele frequencies per se, so it hasn't been incorporated much into modeling. So then then the question becomes: are these duplicate genes that are then becoming polymorphic, in whether they're either gain or loss of function or copy number variation? What percent of the variation in the phenotypes we care about actually map to this versus allelic variation? And that's what we're pursuing. So I'll go through the maze example that we're doing now. And first, we'll do a little brief tour in terms of root phenotyping. So this was a grant that just ended, but we started thinking about how can you study roots in a in a relevant environment. So we started destructively sampling them in various ways and figured out we can get an additional phenotype if we measure the force required to pull them out. And those of you that have done some weeding can probably answer this question: which is harder to pull out, a small root or a big root? Okay, it's like my class. There you go. So in fact, if a hybrid maize, those were inbred lines I showed. That guy Eddie is now an attorney, I think, in Oregon, but he's pretty strong, much stronger than me. And that's an inbred line. So when we started doing hybrids, even Eddie couldn't pull them out. So within this project, this is the Department of Energy, so they love technology. So we engineered and built this system that basically can find an individual maize plant, pull it out of the ground, and record the force. It's also a destructive sampling, so we end up with a root system, a dirty root system that we can clean and phenotype. This thing ended up getting called the Big John because we didn't really know what it was. We've been using it in our own lab and then in collaboration with a number of public and private breeding companies to understand variation in roots. On the right here is basically the trait we're measuring. We grab a plant, we pull it, and we can measure the peak force required for it to rip out of the ground. And then there's another trait, we don't actually understand what it is at all, but it's basically how much that stretches before it comes out. That those two traits are not are correlated, but not the same thing. So, and then there's probably no maize people in the audience here because it's Oregon, but our trait is not correlated with flowering time. That's what is shown in this. DTA is days to anthesis. Every time there's a maize person somewhere, they're like, aren't the ones that flower later harder to pull out? No, they're not. You can all tell them that. So we did this is the international breeding program, CIMMYT. They breed maize and wheat and some other things. So this is from their breeding program where we phenotype this material in Colorado. We're back to our reaction norm plots with wet and dry conditions showing variation in root investment. And so we resequence all of these lines and are making these genome objects that contain presence absence variation as well as allelic variation. And this is, so CIMMYT is the breeding program out of Mexico in Nairobi. And then this is also in collaboration with the Salk Institute, where we're doing some detailed phenotyping as well as the genome assemblies. And as I mentioned, when we can pull these roots out and then wash them and image them, they're complex structures, right? It's hard to know how to describe this. Pulling gives you one phenotype, weighing it gives you another. And then if you image it and extract features, you basically get an infinite number of slightly correlated phenotypes that describe the shape. And so part of what our statistical genetics collaborators are doing is trying to, this old problem of how do you describe the genetics of shape. That's not really anything I'm obsessed with, but throwing it out there. But yeah, we're using this then to ask this question about, so I showed you in maize, we have all of this, there's a whole genome duplication along the speciation event of maize, and followed by loss of almost all of it, but that's all segregating in the material now. So when we make these crosses, we have that. So this is a project, ongoing project funded by NSF. This is my collaborator, Sean Sen, who's the statistical geneticist. And some of the phenotypes we're looking at are expression, in part because those are easy to get, and because we can make these predictions of phenotype from the transcription factors for those. So that, whereas for root phenotypes, we don't really have any predictive idea of what gene models would cause what phenotypes. But we're doing that as well. So we made this cross between two different maize genotypes that don't have as much structural and copy number variation as I showed you. We're actually the strategy is to minimize the amount so that it's not everywhere in the genome and correlated with every SNP, because we're trying to fit those separately in the model. Yeah, and then in maize, you can make these doubled haploid lines. So it's basically like recombinant inbred lines. We're creating this, what we're calling pan-genome aware genotype matrix. This is the phenotypic distribution of that population. Again, wet and dry reaction norms. So this is from, with our Big John pulling machine, we were able to get lots and lots of phenotypic samples within a season. Within the same season, basically get really high resolution mapping of these low heritability traits, some of which we think we know that it's due to copy number variation, but we're still working on proving that. So that's basically what most of what I've been doing the last 5 years or so is putting out lots of populations of segregating lines and measuring their roots. We'll move on to a slightly more depressing note. That's not really the title, but this is my new project that isn't happening. So we started using this same approach to, and sort of the same concept of conditional dependence of allelic or genetic effects on phenotype to look at variation in how maize genotypes, in this case, commercially available hybrids, respond to nitrogen. So, the Haber-Bosch reaction allowed farmers to apply massive quantities of nitrogen to their fields, and then breeders started adjusting the genetics of crops. And so this is basically the target environment where breeding is taking place. And then, so you're maximizing directional selection on yield in this part of the curve, and no phenotyping is done on the rest of this curve. So that it's evolving in a correlated manner or somewhat neutrally. So you see that all these lines, all these hybrids are very high yielding, we give a lot of nitrogen. They all have a monotonic decay, but the shape of those curves differs, right? The parameters describing those curves differ. So if you actually wanted to get farmers to grow corn with less nitrogen, the genetics are already there. They were bred by chance in this breeding scheme. And we showed that this on the y-axis here is basically a parameter describing the curve on the left, and that, not too surprising, but genotypes that have more roots are able to get more nitrogen out of low nitrogen environments. I've led this proposal to basically try to explain to the Department of Energy what genetic drift was, and that there is this variation that could be exploited. This plot on the right is basically saying that the sort of mechanisms, including these extended phenotypes of how there's some microbial interactions with this massive nitrogen environment, and at high levels they cause it to go away and turn into nitrous oxide, and at low levels you select for a community that actually can fix nitrogen. So anyway, we got this grant awarded, and then it's frozen still. So I'm not sure what we're doing when we're doing it, but I'm going to have more free time this summer than I was planning on to analyze the data we already have. So we're doing a lot of stuff with trying to have applied effects in breeding programs, working with public and private breeding programs, but also trying to, within these systems, ask evolutionary questions about the genetics of adaptation. There's a lot of learning still needed, conceptual shifts in the breeding community. The idea that you could find single genes to transform adaptation is still rampant, because if it worked, it would be great, right? It's a lot easier than polygenic adaptation. Part of the idea is there probably, maybe single large effect mutations are out there, but they're really rare. And so if we can move from testing a few genotypes, a few mutations a year, to hundreds of mutations a year, we could find those. And so try to work with people that know what they're doing to create edits that target genes. And then this plug in the middle here is for people interested in theory and implementation of, we still don't really have a clear path of how to add the best practice to add this pan-genome information of how different individuals within a species differ in the not just at alleles, but in the presence of absence of single genes or whole clusters of genes, and do the genotype, phenotype, statistical genetics with that. I will end there and be happy to take any questions. These are my funding sources and collaborators. Thank you. Yes.
So it's fairly straightforward to code that use these genomic variation elements into zero. Big thing that we're discussing now, and that gets to this theory part, is how best to do that analysis and what to measure. We know what to measure, but how do we relate that to population genetics? Things like high, all these kind of things. Those those are all different types. So we're not really, so we're response here. Finding this middle ground.
Yeah, so just by, yeah, it turns out Genomes D works pretty good. When you're confounding paralogs with alleles. So these sort of balancing selection approaches at the population genetics level. When we zoom into those, like in Arabidopsis, like Joy Bergelson and others showed that they had this variation in R genes, and there's papers from 20 years ago in Science about balancing selection at deep coalescent events. That actually, those things that's actually presence absence variation, and they were stacking the alleles on top of each other by accident from different paralogs, and it gives you this deep time. So yeah, so that, obviously doing it in a confused way isn't good, but that statistic, that parameter is showing up for us in these type of approaches. But obviously designing something from first principles, I think would be better, right? But I think in that context, that the sort of whole, obsession with balancing selection in population genetics is in part because there was empirical support for it, but that empirical support is actually turning into copy number variation and not allelic variation. So, so those types of approaches, I think, but yeah, and some of this in maize, for example, is most of this is a single birth of a whole genome duplication event followed by loss, but there's also this ongoing birth, much more than was originally anticipated. So the time part, I think, becomes much more confusing than allelic variation, right? Yes.
So along those same lines, how are you saying that due to statistical genetics relatively sort of reasonable details of what was going on, found this presence absence of transcription factor, and then going back into now, so are you taking markers that are in LD with the presence absence period to add markers back? How are you adding markers back there for actually doing that?
PCR and Sanger sequencing for that example of the.
Assuming that they're in the background, being present or absent at this transcription.
Yeah, so in that case, we are mapping presence absence of the SNPs exactly across there. So, yep. Yeah. So yeah, that in that case, and for whatever reason, the maize community made up their own variant calling pipelines, and if you look at the genome objects they created for all this association, they have tons of hets, even though they're inbred lines. It's like people just kept going. They're like, oh, that's normal. Plenty of hets in my common inbred lines. So we now actually, if you just look for QTL or GWAS hits, where it basically shows up as a dominance phenotype, almost in every case, we found that is due to paralogs getting stacked on top of each other in their analysis platform. In Arabidopsis and obviously, other GATK systems, the hets go away, right, in the software, and nobody looks at them. But yeah, in the crop research, nobody bothered deleting all the hets out of their genotype matrices. So it's actually, it's our main smoking gun at this point of a copy number variation. Yes.
So following on this line of questioning, so it seems like it's even potentially more complicated where instead of thinking about the addition of allelic variation and copy number, that nested sets of inversions, smaller versions, alleles that when present, genomic region that's present could also contribute. So there can be allelic variation just in the subset of genomes that are, so I don't know if you can think about nested sets.
Yeah. So in these crops, people have made all of these nested populations or multi-parent populations, right? So we have, like I said, the phenotype data exists. If we remake the genome objects, you can basically segregate either presence or absence in alleles and biparental crosses, and then in larger association studies look at the cluster effect. There's always going to be a linkage problem, that these aren't independent, right? And you can't break up allelic combinations in an inversion. But there's enough snapshots of that segregating that we think we can, like I said, the idea is to try to write down what % of the variance is due to the allelic variation and what is due to actual presence, absence, or copy number. And you kind of there's a the statistical part is there's a sweet spot of basically where those can be independent depending on the population you're using. Okay. I forgot to turn the microphone on.